All right, good afternoon, everyone. We do a lot of wonderful things here at MIT. We do not control the weather. And so thank you to all of you who traveled through quite frigid temperatures, meaning further than just coming downstairs from the 5th floor in order to be here. It's really wonderful to see so many friends of the department who have come for this very special occasion to be able to mark the 2025 iteration of the Daniel Wang Lecture. This lecture series was established in 1999 to acknowledge the enabling technologies and developments that have sustained the growth of biotechnology and life sciences. It is the intent of this lectureship to recognize and honor achievements on the, quote, frontiers of biotechnology, as it was first named, and the distinguished scientists and engineers responsible for them. This lecture was renamed from the Frontiers of Biotechnology lecture to the Daniel I.C. Wang Lecture on the Frontiers of Biotechnology and Chemical Engineering in 2014, and that was to honor Institute Professor Daniel I.C. Wang, a pioneer and leader in the field of biochemical and biological engineering. Professor Daniel I.C. Wang received the SB in 1959 and the SM in 1961 from MIT, and his PhD in chemical engineering from the University of Pennsylvania in 1963. He then spent 2 years in the U.S. Army at the U.S. Army Biological Laboratories at Fort Detrick, Maryland, before joining the MIT faculty in 1965. And there's our picture of Danny during his Army years. Professor Wang founded the Biotechnology Process Engineering Center, also known as BPEC, through the NSF Engineering Research Center Initiative, and acted as its director from 1985 to 1998. His summer short course, Fermentation Technology, continues to be the longest running course in the portfolio. He was named an Institute Professor of Chemical Engineering at MIT in 1996. In 2000, Professor Wang was given the Temasek Professorship at the National University of Singapore, where he devoted part of his time to help Singapore in their biomedical science research and development. He received many honors, including the Marvin J. Johnson Award and the David Perlman Memorial Lecture, both from the American Chemical Society, and the Food, Pharmaceutical, and Bioengineering, or Division 15 Award, the Institute Lecturer, and the William H. Walker Award from AICHE. He received the Amgen Award in Biochemical Engineering from the Engineering Conferences Foundation, and also received the Asia Pacific Biochemical Engineering Award. He was elected to the American Academy of Arts and Sciences, the National Academy of Engineering, the American Institute of Medical and Biological Engineering, Academia Sinica in the Republic of China, and the International Institute of Biotechnology. Danny consulted for over 50 companies worldwide and was on the board of directors and scientific advisory boards on a number of both public and private companies. He authored and co-authored 5 books, more than 230 publications, and several patents. And the last picture we have there of Danny is towards the later years, and you can see him there with, actually, I can't actually quite see who's there. Oh, with, there's Charlie in the front as well. Before I go on to introduce today's speaker, I want to recognize the sponsor of this award, who is our very own alumnus, Noubar Fayan. Noubar entered biotechnology during its emergence as an academic field and industry, completing his doctoral work in biochemical engineering at MIT in 1987. During his career as an inventor, entrepreneur, and CEO, Noubar has co-founded and helped build over 50 life science and technology startups, and these companies include Moderna, which you might have maybe heard about in the last few years, as well as many others. He's written numerous scientific publications. He's an inventor on more than 80 patents. Noubar is a lecturer at Harvard Business School, and from 2000 to 2016, he was a senior lecturer at MIT's Sloan School of Management. He teaches and speaks around the world on topics ranging from entrepreneurship, innovation, and economic development to biological engineering, new medicines, and renewable energy. He's a current member of the MIT Corporation and a past member of our department's visiting committee. And now, it is my distinct pleasure to introduce today's Wang lecturer, Chetan Goudar. Chetan Goudar leads Amgen's manufacturing and clinical supply function, and he's responsible for clinical and commercial manufacturing operations, new technology intro manufacturing, and global supply of clinical trial material. His 26-year biopharmaceutical industry career has included process development and manufacturing roles at Bayer Healthcare and Amgen, and leadership of drug substance, attribute sciences, and manufacturing functions in science of a polymodal portfolio. These efforts have enabled over 75 investigational new drug applications and 20 commercial product launches. He's a registered professional engineer in the state of California and a fellow of the American Institute for Medical and Biological Engineering. He earned a PhD in chemical and biological engineering from the Michael Smith Laboratories at the University of British Columbia, and he is a grandson of MIT, his advisor being Jamie Perret, who was a student of Danny Wang's. Please join me in welcoming this year's 2025 Daniel I.C. Wang Lecturer, Dr. Chetan Goudar.
Thank you very much. Hopefully you can hear me okay. Yeah, it's really a privilege here for me to be with all of you to celebrate the legacy of Professor Wang. I mean, his contributions to the field, both through the students that he trained and the work that came out of his labs, I think truly is monumental. And some of what I'll share with you, I think, will be a reflection of the contributions that you'll see permeate through from the work that Professor Wang did. And to add to what Chris said, when I joined Amgen about 13 years ago, the head of process development was Jim Thomas, who also was a postdoc here with Professor Wang. So I have a couple connections there to the work here. I want to start by sharing that the topic I'll talk about is the incredible amount of hard work that goes on in translating a wonderful idea for it to be a therapeutic product in a box that a patient can take. We have often heard of these wonderful breakthrough innovations. They'll always be super, super important, but a lot of hard work happens to translate that concept and that molecule into a medicine. And I think chemical engineers play a very, very important role in that translation. And I'll give you a flavor of that. I'll talk a little bit about innovation. But more importantly, or most importantly, I think it's really about service. You know, when we say every patient, every time, it's an ethos. When you're making a decision, when you're trying to decide between A and B, the first question we ask is, does it impact supply? And if the answer is yes, we just walk away from it. So I'll just walk you through that ethos as well, both from how innovation can enable that, but more importantly, the culture and the systems that you need to be able to have that as a way of working. And to start with, I do want to acknowledge many people, because drug development is a team sport. It doesn't matter how much expertise you have in any one discipline. That's obviously very, very important. But it takes a cross-function of expertise and people to really get a medicine to patients. And I think in that team sport, obviously, the work that Professor Wang did, especially in the 80s here on mammalian cell culture, I think have laid the foundation for what we use today in many ways. And the contributions from this department over the last 7 decades has truly shaped the practice of modern biochemical engineering. I mean, that's at least the field I'm familiar with. I'm sure it transcends beyond that. But I think for those of you students, I think there is such a tremendous amount of history and contributions and scholarship in the department that I truly feel very indebted to what I've learned from so many of you in the audience today. And then also to Flagship, the contributions they have made to honor Professor Wang's legacy, and also the billions of people they've impacted through Moderna, for instance. I'm very grateful for the company I work with now, and have many collaborators in the room as well, and then also to the community. It truly, really takes all of us in this journey to make a difference in the lives of millions and billions of patients. So I just want to start by acknowledging your contributions. Your work really matters. You may not really realize that sometimes when you're in the middle of it, but I want to assure you that we learn from what you do, and it really does make a difference. So I have a fairly simple set of topics I'd like to cover. I'll start off building on the theme of a team sport. I'll just talk a little bit about the integrated enterprise. So for those of you who are planning to start companies, or those of you who want to enter this biotech industry, I'll give you a sense of how things fit in. And then I'll talk about the two main things. I'll give you an example of our innovation ecosystem. I mean, it's one way to innovate. but I'll maybe share the ethos and then the so what in terms of business outcomes of how we approach innovation with. And then equally important, I think, is the culture that goes with that innovation ethos that really helps us to meet our expectations of supplying every patient every time. And then I'll just end with a few closing remarks. So on the topic of integration, I'll start with a small story that was the inception of our company and the impact that it's had. If you go back 36 or so years, about a month before, sometime in early May of 1989, Amgen was informed that by the FDA that we would get approval for Epogen, our first product. What we were told is you'll get approval, but the label will change. That's all we were communicated. We didn't know what would be in the label, but what would the change would be in the label, but there would be a change. And then the common dogma was, you can take about a month post-approval to launch your product. That's how people used to think back in the day. But then the approach our CEO at the time, Gordon Binder, took was, he said, no, we have to launch the product within 48 hours of approval. It was not up for discussion. That was just the way it was. And then to make that happen, what the company did was, obviously we were a very small company back then, the product was packed in boxes. They were all in the warehouse. Labels were not printed. And a local printer was on standby to print the labels once we knew what the change was. And then it turns out a day before, we got notification that either an A had to become a the, or a the had to become an A on the label. Nobody knows what the change actually is, but that was the change. And then the minute that notification came, the printer printed them, and then every one of the 650 or so staff volunteered to stick the label on the boxes, and many had to be turned away. But then net, that happened overnight, and the next morning an Amgen employee drove to UCLA hospital with Epogen in the back of the truck. I think that's how we started, and if you come to the company today, that's exactly how we work. It's this collegial, we work in teams, there's this can-do attitude, and we put supply and serving patients first. And I didn't know of this story until I read the book a few years back, and it was fascinating for me to see that if you told the story to someone today, it would resonate very well. Again, it goes back to, if we say something, we do it, and I think serving patients, I think, is very high on that list. A little bit about where we are today. There's a lot of numbers here, but to me, the bottom line here is last year, we were able to reach about 15 million patients, and many of these with, grievous illnesses around the world in different indications, different diseases. We have about 38 commercial products, and we are approved in more than 100 different countries. So that's kind of been the journey from that June 1st, 1989 to where we are today. And then building upon what I said earlier, we've all had these stories of these brilliant ideas, this great target, this great molecule. All of that's obviously very, very important. But for you to be able to go from that breakthrough idea into a medicine, what really matters, the end game here, is a product in a box that's approved by the health authorities to use. That is when you really have an impact. That's when you really, really reach patients. But the path there is anything but linear. And even though we have done it 38 times so far, the 39th time is just as complicated. There is always something different. There is always something new. And I think just an acknowledgement of that reality really helps one approach this art of drug development or commercialization with humility and with resilience. I think Steve Jobs said it really well. There's a lot of, I think he used the word craftsmanship, which absolutely is true, but it's also a lot of blood, sweat, and tears that go into when you have to convert that idea into medicine. And again, a lot of what we do as chemical engineers in this industry, we straddle the innovation, and then it's our job to take that idea and innovation and translate that into a process, then into manufacturing, and then into a product that can then be sent out. And then maybe just a little bit of clarity, right, on that same concept. I come from this function that is called technical operations or operations. Different companies call it different, with different names. But what we do, our job is to take this breakthrough innovation, the molecule that comes from our colleagues in R&D, and to translate that to a medicine in a box that you just don't launch with, but that you have the ability to supply reliably for the next 20, 30 years. Consistent quality, highest levels of compliance, and assurance of supply also are part of that equation. It's not just the initial launch. And then when you do that well, then you enable your commercial colleagues to then go and interact with healthcare providers and so forth to have patients get access. The key thing here is the intersections. You'll be surprised to see that in some organizations, these are 3 circles with no overlap. They may look this way on a slide, but in reality, the intersections are either non-existent or very slim. And I think that's a lost opportunity. If you work in silos, again, all well-intentioned, but if you don't have empathy for what this group does to take an idea and make it into a molecule, it's easy for us to criticize and say, hey, this molecule is not manufacturable, or it's difficult to make, what were you thinking? You can have those types of reactions, but it doesn't really help. Because what if it's a really complex biology? They can't prosecute with your preferred modality. They have to go and find something complex. Our job is to share that feedback, of course, but there will be times when biology dictates what modalities comes to you, and then we have to then be the can-do enabling function that enables that. Same thing here. Our commercial colleagues, will expect, certain margins or certain other competitive advantages in the marketplace. And if you're not empathetic to what they're, seeing and learning on the front lines, you can't build that in to your process design and manufacturing processes. And then what ends up happening is you think you have done the best job you can, but it's just not quite the right thing for your downstream partner. So again, I think if you want to start your own company or if you join an enterprise, I find the most impactful people are the ones who are very fluent at the interfaces. It's funny because sometimes, when we interface here, our R&D colleagues think of us as people from commercial because, and I'm anything but that, right? But just the mere mention of the economics brands you as a commercial person, it's best to take that as a compliment and move on, right? And here, sometimes you get branded as, this R&D person who has no feel for reality. Those things do happen, but the more fluent you get at the interfaces, I'd argue that you can really, really have an impact and then transform how these groups work seamlessly to get a medicine to patients. And then my last point on this topic here is if you just double click on that middle portion, right? As a chemical engineer, you'll most likely start in process development. I have many friends in the audience who have spent a lot of time working in this space. Very, very important, right? What this group does is you take that sequence of an antibody or you take that chemical structure and then you design a process to actually manufacture that. Very, very important. A lot of real awesome innovation happens here. But then, that is not the end of the world, right? Someone has to manufacture it for the clinic. Then if it's successful, then it goes into commercial manufacturing. Somebody has to design and build the plants that actually make the product. Quality and compliance becomes super, super important because you have to have consistent quality. And then obviously, I think, building in the latest approach to digital is super, super important. And then you have to improve. Last but not least, supply chain. I have direct oversight over supply chain now, and it's a very complicated piece when you think of raw materials and tariffs and all of that's playing out today. So it's this ecosystem that's the middle part of an enterprise. And again, you'll probably be very comfortable in this space. You have trained your whole life for this. But my advice to you is try and move along these different functions. At least understand them well. If you can, take roles in different functions. And I think it just will make you a more holistic person, and you will just be better equipped to do your job, no matter what function you are in a significantly better way. later on, right, when I talk about innovation, I'll just make a point here. Our approach to innovation is a little bit top-down in that the people who lead these functions, they get together and decide what is the most impactful thing we should work on from an innovation standpoint. So it's not someone in process development thinking and saying, hey, look, I have this great idea I want to work on. That may be true, but someone from supply chain can come and say, we have a real need for a local manufacturing capability in the Asia-Pacific region. You would never think of such a problem statement sitting in process development because you just are not exposed to that way of thinking. And then the response to that is our traditional ways of manufacturing, building big, expensive manufacturing plants to serve globally, will just not work in a region. So then that problem statement becomes a challenge for these functions to then figure out how do you design and develop a manufacturing solution for their supply chain problem. Right? So again, I think this is a real example, not exactly what I said, but on those lines. And to me, those are the types of discussions we have, which then decide what do we focus innovation on, and those ideas get resourced and funded. So again, that's another advantage of an integrated way of thinking where you don't have a silo deciding. I mean, each function can come up with a problem, but you look at it in that global context. So then I'm just going to transition to the second part, right, where I'll double click on what I just said, the innovation part. The key word here is intentional. We have a lot of creative people who work with us, and obviously everybody has lots of good ideas. And the intention is not to not have staff have the freedom, but the way we take a look at it is what are the most critical business problems that we need to solve. And oftentimes, I think visibility to that is not at the individual staff level. So we start there, and then every year, they don't change every year, but we revisit them every year. You can count them on one hand. So everybody in the company knows what are the most important things for us to transform. These usually are big ideas. They're transformative. They are step changes. And then when those discussions happen, everybody gets to see what those ideas are. And then when you need funding, people, equipment, you don't have roadblocks. Like, for example, if you have a solution that's aligned with an imperative, you don't have to go around asking people for permission or asking people approvals for funding. It happens seamlessly. There are many, many examples where people have really, really good ideas, but they just get stuck because not everybody is bought in, and you just don't get the right kind of funding and resourcing to advance those ideas. Whereas this framework really helps us move things forward. And when that happens, right, you're focused on developing the solution and not trying to navigate organizational complexities, which can be very, very time-consuming and draining if not done well. But then our staff, right, have a tremendous amount of freedom. No, I mean, the people here don't tell you how to solve the problem. They're better off not doing that because I don't think that's going to work very well. But I think we have our teams, who then will come up with the most innovative solutions to those problems that we then move forward. So what have been the outcomes? And I'll give you a couple examples in the next few slides. If you look at next-gen manufacturing, so it's almost a decade now since our first plant in Singapore was approved, and that's a manifestation of the company deciding that investing in that technology, the technology suite, was important for us to maintain a competitive advantage in manufacturing, and that's worked out very, very well for us. If you look at productivity, I'll show some examples. These really are industry-leading productivities that have come out based on a singular focus over almost a decade, both on the biology side and the engineering side of how do we improve the output from our factories when it comes to expressed biologics production. On visual inspection, I know some of you in the audience have had LGO students who have worked on that, right? AI has been a part of our visual inspection process for a long time. And it's had a tremendous impact in the ability to release tens of millions of products to patients in a very efficient way. And then the reliability of supply. We're one of two companies that have not shorted the market in the history of the industry. And I don't think that's by chance. That's very intentional, very purposeful. And my best part, right, is every year we nominate internally these ideas. We have an innovation award where people nominate the best ideas. And I went back and looked at the last 3 years. And what you see there is every award-winning idea is actually implemented in GMP manufacturing or GMP testing. So that just tells you the connectivity between an idea and the implementation. Now, does it constrain? Absolutely it does. But when you see that you worked on something become a reality, it's incredibly inspiring for the staff. And then they'll cycle on to the next. So again, I think I'm not saying this is the perfect way, but a purposeful approach to innovation and defining the solution space really has helped us make meaningful advances in business outcomes. I like this example a lot because you can learn a lot from other industries. And if you look at the semiconductor industry, for a long time they were focused on clock speed, right? Intel would say 3 gigahertz, AMD would say I have a 3.2 gigahertz processor. That went on for a long time. But in about 2005 or so, they kind of hit the power wall. The green squares here are the clock speeds. They realized that when computing was moving to mobile, you had to manage power. The power per watt became important. You just couldn't fit in an infinite amount of power in a battery to power a laptop with those high speeds. And then they had to change the approach. They kind of kept the clock speed constant, and then you had more cores on a chip, and you still got better performance. So that was a breakthrough, and we all benefit from that innovation there. And I think a similar thing happened to bioprocessing around the same time. Constantine is here in the audience with us. But in the 2006 Cell Culture Engineering Conference, right, he proposed this idea of integrated continuous biomanufacturing. The industry had done perfusion cultivation where it was needed in enzymes and blood factors and so forth, but we had not looked at it holistically as a suite of technologies that could enable bioprocess production all the way from bioreactor to purification. So when that happened, I think it's kind of opened up a space, and there's a lot of really outstanding work that's happened across the industry since then. The approach that we have taken is a continuous technology has about 50 things inside of it. So you can think of that as a menu of things that you can pick and choose. And in some cases, we have picked 30 out of 50 things. In some cases, we have picked 4 out of 5 things. And we have built that into our manufacturing capabilities. So this is real data from one of our manufacturing sites. And just walk you through this. If we just look at it as a function of time, in the early 2000s, this was a single product plant. And then over time, as the portfolio diversified, as the portfolio grew, you had more products. So it's truly a multi-product facility today. If you look at the output, this is just kilograms or metric tons of protein being made. It's all mammalian cell culture based proteins coming out from the plant. You see a 5x increase from 06 to 23, and we expect another 10-fold increase in output from the same plant in the next 7 to 8 years. Okay, this is a big plant. So that 10x increase is a very big increase. And it's a very meaningful improvement, because if you don't get those types of outputs, you'd have to go and build 2 or 3 more plants. And these are $1.5 to $2 billion of investments each. This is innovation. Continuous principles have had a very big role to play in that, along with improvements to cell line development and so forth, right? But nonetheless, this is just mostly innovation, with obviously some continuous improvements built in as well. So we do both. But look at the resources piece, right? We were able to halve the number of resources on site from the early days to now, and we expect to stay flat as we increase the capacity or output tenfold. And the translation of that is a very significant reduction in the cost per gram. Again, glycosylated proteins, all of these are glycosylated proteins, antibodies, multispecifics, it's the entire mix of products there. So the point I'm trying to make here is when you have, and obviously this is a big focus for us over the last 10 years or so, is improving productivity without compromising quality, without impacting reliability, and optimizing the economics of the process was a focus. And there's a bunch of innovation that has happened. And we are at a point now where we feel that we can do more in the next 5 to 7 years. Okay, so again, I think an example, a very tangible impact of innovation. The next example I'll give you, right, is maybe very counterintuitive. We are an innovative company, but we also have 8 biosimilars that are commercially approved. And if you go back 15 years, there was a debate. Why are you working on biosimilars? Is that a distraction? Is there an opportunity cost because you are deploying your resources and time on biosimilars? And obviously, it's a good, healthy discussion to have. We made a decision to go into biosimilars, and that's annualizing over $3 billion a year now and is growing. But here's the interesting thing, right? In the early days when I had just joined the company, the perception was that, we can maybe hire some contract workers to help us with the biosimilar portfolio. That just didn't work. They were the most complicated molecules to work on because being better was not an option. You had to be the same. You can't improve on an old molecule. You have to be very comparable. I think we had a good platform of understanding of molecular attributes and the relationship to function and efficacy and safety. But we had to up our game. And very quickly, we had our best people working on biosimilars. I mean, it was a badge of honor to be working on a biosimilar because you were considered more experienced, more sophisticated, more of an expert perhaps, to be able to work on modulating the product quality attributes up or down, left or right, depending upon what the similarity criteria was. So we started off with an innovation base, but our understanding of molecular attributes and modulating them, I think, has gone up by, it's a step function primarily because of biosimilars. Had it not been for that, I don't think we would have pushed ourselves that hard because the bar for comparability within your molecule is rather low compared to similarity for a biosimilar, which comes from a different company, different manufacturing platform, different hosts, and so forth. And then what we have learned, we then channel that back into our innovative portfolio. So comparability these days is not the big scary thing it was a few years back because we have that experience. More importantly, right, there is no two standards. Like I said, our best scientists work on biosimilars. It's the exact same manufacturing plant that makes both types of products. So if there is a box with our label on it, if it's a biosimilar or an innovative molecule, it's the exact same people, process, equipment, plant that do it. So I think from a societal perspective, I think you get the same quality for a fraction of the cost that can also help our healthcare systems as well. So again, I think superficially this may look like a distraction, but I think from where we stand as an operations function, this has been a terrific learning experience and a driver, I would argue, for innovation within our innovative portfolio. The next example I have is platforms. Again, I think when you think of a platform, you may think of efficiency, but I would argue it's an innovation engine as well. So what is a platform, right? And if you go back and look in history, anytime you standardize something, you can think of that as a platform because then you can manufacture things more efficiently, things can fit, and so on and so forth. So there are some really interesting examples across civilization in general. One of the examples I really like is the container. So Malcolm McLean, if you have not heard about him, he was a truck driver in North Carolina who used to carry bales of cotton in his truck to a port in North Carolina to be sent to New Jersey. And what he learned was he had to wait for the cotton to be offloaded from his truck and put on the ship. It took forever. And he just got sick and tired of waiting. And then because they used to have all these different types of cargo, and the time it took to load was almost as much as it took to travel. So it was very, very expensive. So he tried to put trucks on ships. Didn't work very well. And then he came up with this idea of a container on the back of a truck that could then be stacked. And that began the revolution. He borrowed money and bought 2 World War II tankers. It's a fascinating story. Bought 2 World War II tankers. No one believed him at first, and then he told the people, look, people can steal your cargo if it's locked in a container. So that's how we convinced people to try him out, and then the rest is history. Again, a very simple example, right, of an innovation, in this case, of standardizing how you ship cargo. And I would argue that it's saved, or it's helped many, many people, hundreds of millions of people get out of poverty. So Forbes called him one of the few men who changed the world. An example of platforms are efficient, right? So you can see the logic, but what does that mean for us in the biopharmaceutical manufacturing context? So a simple question, right? You have, let's say, a chromatography operation. You have 2 processes, molecule A, molecule B. One of them has a pH of 6.3 for a buffer, one has 6.4. Does it matter? If you ask a scientist on the bench, right, they may say, look, for product B, 6.4 is better. I get an exquisite profile of elution. Look how elegant it is. I get a 0.2% improvement in yield. All of that are very good first principles-based arguments, right? But think of that big plant that I talked to you before, which is making metric tons of product. You have 6 to 8 hours to finish this unit operation before the next product is going to come in. And if I have to switch from pH 6.3 to a 6.4 buffer, I have to make a whole new batch of buffer. This unit operation probably has a half a dozen buffers. If they're all different, it's very, very difficult for me to dump everything, remake all the buffers, and then still maintain success. I mean, our criteria for success rate is 98%. So if you're 97.9% successful, you've failed. When you have that high bar, you can't afford to mess around with a 6.3 and a 6.4 buffer, right? So the context then becomes, this may not be the place for us to innovate. Now, it doesn't mean that we don't innovate. What we tell our scientists is, look, find a better resin that will transform the operation, or go from bind and elute to maybe a flow-through mode, which will speed up the operations, and then we'll update the platform. So every year, the platforms get updated, and then we celebrate that. If you're an inventor, you get to make the case, and you get to be credited with version XYZ of the platform. So the innovation, so in my view, platforms don't constrain innovation. Platforms enable efficient operations. But then you have the freedom to not spend your time in optimizing 6.3 and 6.4, but think of a big idea that can fundamentally transform this unit operation and focus your time and energy in that space. So I see platforms as that foundation on which you enable reliability, efficiency, but then you free up time to go and do something else. And then a similar example, right? In my previous role, analytical methods, you may think, why do you need a platform? It's the same thing. You have a QC lab, which is a GMP lab, and they have to test the products that you make. And there's all kinds of mundane details here, right? So what is the cartridge temperature set point? 25C. Can 20C work? Probably. Do you want to spend your time trying to figure that out? Probably not, right? So we have this level of detail as first intent. You don't have to start here, and if it works, stick with it. If it does not work, you make an exception. The bar is high. And then every change that you make has to be approved by a vice president. So that's the level of stringency that we have around platforms. And again, it was a cultural adjustment for the first couple of years, but now we have moved on to a point where staff understand, acknowledge, and then we focus innovation on some of the bigger ideas. So again, I think biosimilars and platforms, right, are two things that can look mundane and boring on the surface, but I think a couple clicks down, they can truly be enablers of innovation. And the last thing I'll talk about is just the culture. Culture is a topic which we don't talk much about at conferences. We usually talk about technologies and ideas and advances and things like that. But I do want to spend a little bit of time because I think that is indeed a very differentiating dimension. So, I mean, different places are different things too. But for us, we have a very simple way we look at things. It starts with reliability. It basically means ensuring supply takes priority over everything else. So you can be in any discussion, any conversation. You could have a great idea for a next-gen process, and obviously an earlier implementation will be beneficial on all fronts. But if that's going to compromise supply, we just will not do it. Again, it brings clarity to conversations. It helps you focus on what's the most important thing in your journey to get medicines to patients and also to improve. So reliability takes priority. We always want to be better. I think that's just the way it is, right? I mean, things are very competitive, things are evolving, and I think we enable that with innovation. We do continuous improvement as well. I think it's a very important component, but the biggest impact that we have seen is when you truly bring about breakthrough innovation, you really can build in efficiency, and then obviously. Things change, right? I mean, the market dynamics, the geopolitical dynamics today. How quickly can you respond to that is super, super important. And then obviously we want to have the best people. All of these really are in service of our intent to supply every patient every time. And again, I think it's simple, but I cannot tell you how many times there have been conversations where things can go in 10 different places, but when you have an anchor that you come down to, it just makes it very, very clear. We had this very difficult time when the hurricane went through Puerto Rico. Puerto Rico is our largest manufacturing operations. We have more than 3,000 people who work there at the site, and a lot of our products are made there. It was a very, very tough situation. At 250 kilometers per hour winds went through. Some of our plants were impacted in a very, very severe way. In fact, there was no place to even sleep on site. Everything was wet, and we had our leaders were there 24/7. They took turns to be on site, and some of them were sleeping on desks because the carpet was very wet. So it was a very, very challenging time. But then I think the thing that the company did was we made sure our staff were safe. I mean, they didn't have electricity at home. Hot water, a hot meal was a luxury. And the company made sure that the staff were taken care of, and then whatever had to be done. I mean, our drug substance plants ran through the hurricane and beyond because we had active manufacturing ongoing. Those things were not affected, so that much. So those operations continued. But I think what this demonstrated again, right, was that despite all of this, supply takes priority. So that was a very clarifying principle that helped us through this difficult time, coupled with obviously putting employees first, which I'll talk about in the subsequent slides as well. And then we also have a safety stock, like most companies do, that we were able to leverage to make sure that we were able to supply all of our products despite the challenge. So again, I think when everything is going well, you may not be tested. But when you have a catastrophic incident, I think it does put your fundamentals to test. And this was an example of us responding to that type of a situation. Another example, right, of the culture that I feel very privileged to be a part of is empowerment. This is from Gordon Binder's book. He was the CEO of Amgen at that time. And in his book, he has this figure 10.1, which talks about employee independence. And his notion was, in a typical company, who manages you? You're kind of the medium-sized circle, and your boss is the biggest circle. This is probably true in many, many places. And then obviously you have a peer who is a smaller circle. And his representation, which I think is very accurate even today, is you are the biggest circle. And more importantly, your boss is the smallest circle. And I find it very true, but also very empowering. Because what it says is the product team that you're a part of, the product is way more important. The mission is way more important than your function, your reporting line, your silo, or whatever that may be. And again, I think it seems simple, or it seems, 6 circles. But when you look at it and how it manifests itself in day-to-day operations, discussions, decisions, and meetings, it is a very different way of working. And I find it to be very, very impactful. This line is from the same book from Dennis Fenton in 1981. We were formed in 1980. He was the first head of operations. He came from a big company. And he said he actually was heard. Just imagine, right? We hire people trained by MIT. And if you're told what to do, it's a complete lost opportunity versus asking you, how do you think we should solve the problem? And 9.5 out of 10 times, you get a better answer asking you how we should solve it. Seems very simple, but I can guarantee you, it does not happen as often as it should. And this line actually caught me thinking, how many times have I made that same mistake? And the answer is more than I should have. Because you sometimes have the perception that because you've been doing this for some time, you tend to know the answer. You may have an answer, but it's almost not the best answer. And being open to new ideas, newly trained professionals, I think is really, really important. I think we do a pretty good job, but for us to go back to the origins of the company, to have a head of operations express the sentiment, I think was special. So I do think it's something that we try and do, and it's very, very important. Otherwise, you're missing out on leveraging the collective wisdom. The other thing which we also try and do is doing hard things the human way. If you look at drug development, it's a very complicated thing. It's a very difficult thing that you do. And you can get frustrated. You can be in a rush to make choices, and you can forget that you have people on the other side. And this is a framework that we have used for a few years, and it's worked very well for us. What it basically says is there is a wisdom axis, and then there is a compassion indifference axis. We don't really have this bottom left problem where you don't know and you don't care. Such people usually don't end up saying, I want to come and serve patients. That's not really a problem that we have. But this is a real problem for us in that you have so much empathy, which is a good thing, that you just are not willing to ask the hard questions. It's coming from a good place, good intention, but you're not really solving the tough problem. So that's a problem we still have. And we also have this problem where you just want to get it done. It doesn't mean you have a terrible intent or you're a horrible human being, but you're just focused on trying to get things done that you forget that there has to be some compassion on the other side. Whereas on the top right is where you really want to be, where you don't shy away from the difficult problem, but you acknowledge there is a human on the other side. And I've had people tell me in meetings, you're in box 4, move up, right? And this can be anybody in the room. You're empowered to say that, and in fact, we thank you for the feedback, and I correct my behavior. And it's been a very simple thing, because like I said, we really don't have a box 3 problem, but we have a 1 and a 4 problem. Again, very simple, But I think just keeping front and center that we'll not shy away from the difficult decisions, the difficult problems, but we'll do it in a humane way, I think has really helped us bring people along and make these tough choices without having people feel like they've not been heard or they've been dismissed or assumptions have been made. So again, a simple concept, right? But it goes a long way, I think, in being effective. We'll talk about safety, right? I mean, this is enterprise-wide safety performance data, not just in the lab, but even including our sales force who drive cars to go and meet with the doctors and hospitals and so forth. So we were not doing all that well. That was like median performance, and we wanted to kind of be where the best in class are. And then what happened in 2018 was we moved to an actively caring culture. So what does that mean? When you see someone texting and walking, it happens a lot. You can approach that in a punitive way or say, look, hey, don't you realize it's not a safe thing to do? That can be one way to approach them and make them feel like, hey, they're not doing the right thing. Actively caring is, hey, look, I'm concerned about your safety. I want you to go home safe. Can you please not text and walk? It's a shift in the narrative, and it's not just when you see someone texting and walking. It's in your labs. It's in your workplace. You just demonstrating that, look, it's not just an edict I want you to follow, but I'm really interested in you being safe and the person coming after you being safe. It takes some effort to make that transition, but once we did that, you see the performance that follows. Again, this is across 28,000 people. This is not just one lab or one building. This is the collective enterprise where we made a change in moving to an actively caring culture, and I think it's really, really been helpful. Also, when it comes to work, right, we don't have any rules on how many days you need to be in the office. People have the flexibility to decide what is best for them. So again, I think that's an empowerment piece. I'll talk about AI, right? I just have one slide, but I want to give you a couple examples of AI, and then we can transition to the last part. I don't need to tell you, but AI will transform the entire business of making medicines. There is no question. It's not an if, it's a when, and it's a how, and it's as simple as that. There are a couple things which I think will be very, very important. You may see a massive advance in technology in 3 months or so. We will not be that fast. The kinetics of adoption will significantly lag the kinetics of technology acceleration. We just have to accept that. I mean, it may feel, like disappointing or frustrating, but that's just the reality. And the second thing is we'll have to have human in the loop. So I'll talk about 3 examples of using AI in the regulated space. Again, right, the first instinct one may have is, look, you can't really use AI in a GMP setting. Again, it's a logical response, but I'd argue the reverse is true. Because a GMP environment helps you constrain the problem, helps you constrain the solution space, and you end up with better outcomes. Top left, right, health authority questions. 38 products, 100 countries, and we're constantly improving, which means we are filing more than one file every single day in some jurisdiction. You're constantly filing for a change. And you know what? You get questions back. Hey, what about this potency method, and what about this condition? Can you imagine how many times that question may have been asked in the last 20 years across how many products? You can't keep track. We have those people who remember in 2014, the Czech Republic asked us this question, right? We have a few of those people, but they won't be around forever. And what we have done is we've taken all that information, and now we have a large language model sitting on top of that. So it has got access to those questions, our responses, the jurisdiction, and the outcomes. And when you get a new question, if you ask the same question to the large language model that has access, constrained access, okay? It's not responding, it's not thinking, it's not doing its own thing. It's working in that constrained space. Its first response is just as good as, or even better, than what your SME can come up with, because it will contextualize that question against every single response in the past. So not only are you getting a response, you're getting the context across that question, jurisdiction, products, modality, and all of that. So an example where you can do, again, but the response we submit is not the response we get from the large language model. That's, again, right, so that's the human in the loop component. There is, you get a start. And then we have the same subject matter experts now armed with this output to go in to prosecute that, to do whatever they have to do to be convinced that they're data verifying and signing off on a response that meets the highest quality expectation. So it's a tool, it's not the complete solution. Second example, right? I mean, we use a lot of single-use systems. And if you look at the amount of, we have thousands of raw materials that make up, a piece of tubing is a separate SKU. And let's say, you have a need to build a new single-use system because the product needs something different. That becomes a whole new part, which can take you months to get the manufacturer to make it, test it. It has to be a part of your SAP system, specifications, release, all of the boring stuff, right? So that's one way to do it. Or if you train a large language model to know about all of the components that you have, also have it look and see what you have in inventory. And then you ask it to say, look, I have this need. Can you build me something with the existing components that I have, with the inventory that I have? You will get an answer that is usable. It may not be the most elegant because it is trying to do what it can, but it's again a starting point. And then you can improve on that, and then you can get your quick solution while you design your ideal solution down the road. Another example, right, of you leveraging AI. And my favorite is the third one. We all have manufacturing deviations. That's just the nature of the game. And then when you have a deviation, the first thing you do is you have to triage the deviation, right? I mean, is it a major deviation, minor deviation? Some companies have numbers and tiers and so forth. But you have SOPs that govern exactly how that works. This is not subjective. There is a set of sequence of questions you answer that leads you to the outcome. And lo and behold, right, if you train a large language model with your SOPs and your past history of how you have triaged deviations, the output that you get when you have a real deviation is almost as good, if not better, than what a human can generate. And the best part is it is very consistent because it forces you to go through every single step that we have in the SOPs, as opposed to, as an expert, I may gloss over a couple obvious things that may not really be that material to that particular situation. The large language model has none of that. It walks you through every single step, and when it drafts the report, it has all of that context built in. So the end product has all of that detail, and then most importantly, it is still the subject matter expert who reviews, verifies, and signs off. So again, right, I mean, you've all seen the innovative side of AI, but I would argue that antibody discovery, obviously there's a whole lot of things that can be done there. Supply chain, yes. Commercial, yes. But even in a GMP environment, I think the constraints are our friend. And that's what helps you get to a good solution. So I just want to leave you with, it will transform, but it'll take a little bit of time, effort, and patience before we get to see the full potential. So maybe I'll just try and wrap things up, right? I think in most companies, I think science and culture defines the DNA. But I think there is a third dimension. I call it systems, you can call it whatever, right? I mean, it is that we will innovate based upon what we think are the most critical business outcomes. That's an example of a system. We will use platforms to be efficient and reliable, but will innovate to build new platforms. So to me, those are some examples, I think, of things you can do. Because if you want to scale, and if you want to be doing this for 10, 20, 30, 40, 50, 100 years, you will have to think a little bit different, and you'll have to build systems in place that will help you stay competitive for the long run. So that's maybe one thing just to keep in mind. I do want to maybe share a couple remarks, right? I mean, Charlie's here in the audience. And going back to Professor Wang's legacy, Charlie talks about the impact Professor Wang has had on the industry. I think it is, billions of patients have benefited from the innovation that has come either from Professor Wang's students or from the research that has come out of his lab. So it's a staggering impact when you look at it in the overall context of the contributions. And then tough yet caring is that attribute, right? I was just talking to Wei Shu last night, and he said Professor Wang was tough on his students, but he was tougher on himself than on his students, was something that Wei Shu said last night. And then he also mentioned the KISS principle, keep it simple, stupid. So I think that attribute here, right, is tough but caring, is something which I feel is a very impactful way for one to just conduct oneself. This is a short video. So that looks like a playful 3-year-old with a backpack. I mean, that's what it looks like, right? But it's much more than that, So that patient has acute lymphoblastic lymphoma, horrible disease. And if you look at the standard of care, this is overall survival. It's about 66%. And this is chemotherapy, and some of them also have to have bone marrow transplants as well. So it's a very, very terrible disease. And then with Blincyto, right, which is a bispecific T-cell engager, the overall survival goes up to almost 93%. One example, right, of hundreds of examples across the industry where we, I think, as a community have developed products that make a real difference in people's lives. The backpack actually has a pump because this is CIV administered. This is our first generation bispecific T-cell engager. So that child is carrying the pump in the backpack. It's a 27-day administration CIV. So to me, that's what it ultimately comes down to. When I reflect upon the legacy of Professor Wang and many of you in the room, you have made contributions that make that outcome possible. Sometimes directly, but sometimes from a distance. It doesn't matter though. It is still a very tangible, a very impactful contribution that you have made and that you will make as some of the students go into the workforce. And just keep in mind, right, that you have an opportunity to really make a difference and save people's lives. And to me, when we say every patient, every time, it is that call to serve, that call to service that transcends everything. And I hope, that even if I can convince a couple of you considering a career in quant trading or something, to come and join us in this journey to make a difference in people's lives, I think I'll be very happy. Thank you again for the opportunity, and I'll be happy to take questions.
Questions?
You have this ethos, you have the ethos you described. How far back does that date? Does it go to the origin of the company? And then how has the company maintained it in the face of stressors and crises and leadership changes? You have some continuity of CEOs, maybe, which helps. But how do you see that being so sustained?
Yeah, so I think in the early days, I don't think we had defined it quite as such. This every patient, every time goes back a couple decades. So I think you can say the formality of that is maybe halfway through our journey. We've had CEOs with a decades plus tenure, which certainly has helped. But for operations, I think supply has always been front and center in some way, shape, or form. And whoever has succeeded the previous person, it's just been there. And more importantly than it being there, when you see it play out in a real situation, I find that to be the most compelling reinforcer, because you can have things on a wall, you can have things on a slide, but when you have a real difficult situation with real consequences, when you still choose to say, look, what matters to me is that option, while attractive for whatever reason, will risk supply, I will not do it, and there is no discussion. I think to me, more than anything else, that is probably the reason why it has sustained, because you see that practiced over and over again.
Thank you for that really nice talk. I'm curious, especially given you give this example of the bispecifics, you're making more and more complex molecules. Where is the frontier for making these drugs really accessible? Is it on the production side? Is it on the purification? Is it something else?
Sure. I think when you talk about the more complex molecules, expression is still an unsolved problem. I do feel like we have come a distance in expressing antibodies, but when you have these bi- and trispecifics, we certainly feel there is improvement in expression. I think that's probably the single biggest lever that we have. And also consistency and quality, because when you have many chains, sometimes you make like a half of a molecule or some normal combination of a molecule that you have to then purify out, and you lose yield as well. Correctly expressed molecules, expressed at a higher level, to me would be the single most important factor in those types of modalities. And that's the work that a couple of you are doing. So I'm very optimistic about what comes out of that.
I'd like to ask a question that's an extension of Katie's. As biochemical engineers, we've gotten to be really good about controlling the performance of the reactor. Mm-hmm. But that's only because the challenge of controlling the performance of the cell, which is doing the job. And one of the areas I know you have been very active in is thinking about how to better control the real job to be done, and that is the cell. Could you spend just a moment and hit a highlight or two about how the innovation on controlling the cell, the biology, versus what we do as chemical engineers, easily the reactor?
Yeah, great question, Charlie. So when we sought to make the next host, if you will, that's a very rare event in the industry, right? Because we typically have a host that we have used for a long time. And when we sought to make a new host about 10 or 12 years back, that's exactly where we went to and asked ourselves, right? What are all the things that are bottlenecks today? Not just, increased expression is one thing, but in terms of raw materials, for example, right? Insulin is an expensive raw material, or growth factors. How do we find a way to work around that when you build a new host, for instance? And then, we obviously were moving towards continuous manufacturing, which meant you had to have filters in the process and so forth. And how do you select for a host that is very facile in working with going through a filter and coming back as well? And then, how do you select a host which probably has a less propensity to produce lactate, for example, and so forth? So we asked ourselves those questions, and we spent about 2.5 years just trying to adapt this new host through these different sets of conditions. And it took us some time, but what came at the end of that was a very hardy host, which was used to the conditions that we would expose it to later. So that's maybe part A of the problem. And the part B was actually the molecular biology itself. How do we design vectors? Obviously, lots of work has happened in the past 2 decades as well. So that was the other place where our molecular biologists did some really nice work to optimize the vectors that went in format specific, because the vectors that worked for a 2-chain antibody are different when you have multiple chains and so forth as well, right? So I would say it's in those 2 dimensions that we worked on to optimize the inherent ability, if you will, or familiarize the cell with what it's going to see in real life, and then do the molecular biology on top. And it's the combination, I think, that's been fruitful.
Well, we're going to cut off the questions in the interest of time, although our speaker will be here for a few minutes afterwards. I have to go to this side and then come back to the other side, so I'm rambling just to fill the space. But I want to start by saying thank you for coming to visit us and for talking not just about the importance of technical contributions, but how you do the work matters. And I wrote down something you said very early to our students, which is your work really matters. And so I hope, especially for the students and postdocs, you've been able to think about not just how your technical contributions can lead to life-saving medicines, but also the way in which you do your work both here and as you leave here can contribute to the culture that allows for those innovations to continue. I left out one thing in my introduction earlier today, which is that it is our speaker's birthday. And so we're not going to sing.
Thank God.
But I do hope that as you have given the gift of your time to us, that you have felt that the time you've had with our faculty and with our students has been a gift to you as well.
Been
Thank
great.
you so
Thank
much
you.
for coming,
Thank
and
you.
happy birthday.
Thank you, Chris.